So thank you so much for the presentation. And to start right away, well, here I put in red my province in Argentina so that you know where I'm based. About treatment for cutaneous leishmaniasis. Current therapy is, as Byron already explained, mainly very toxic, and most of the complications coming from this therapy arise from that, from the toxicity. The most useful, which is oral miltefosine, has this main disadvantage that you cannot use it in children, you cannot use it in women of childbearing age without concurrent things not to get pregnant. We are in dire need of something new. And I really like the idea of topical treatment because, well, it has some obvious advantages. We will talk about the disadvantages later. So mainly the advantages are derived from the low toxicity because the drug would only work at the site mainly. It would also enhance patient compliance. It will be easy to use. It will have decreased side effects, and it will be useful in a context with low medical infrastructure, which is most of the regions where the disease is endemic. And also, since before they were talking about the amount of children that are affected by a visceral leishmaniasis. In cutaneous leishmaniasis, also children can be up to 70% of the patients, depending on the region. So it is really important to be able to have a treatment that is good enough for children and women. So where are we right now? This that I explain now, it's been obvious for many years. There has been a lot of work done on trying to develop a topical treatment for cutaneous leishmaniasis. However, there is only one, and I put here paromomycin, but I hear somebody before said that it is not commercially available. So maybe I should also cross it with red, like Byron said. Paromomycin is not even commercially available, and right now there are, as I know, only 2 topical formulations in clinical trials, one of paromomycin and gentamicin, and then there is a liposomal amphotericin B that has gone through the first phase of a clinical trial. So it's really scarce what the amount of new things that are there, right? There are some new topical formulations with preclinical data available. Most of them have been published in the last couple of years, but of course only on mice, and most of them use miltefosine, which has proven to be very effective, but the literature has a very so each paper has worked with only 1 or 2 variants of leishmania, which is we know that the different species react differently with to the drugs, so it is really important to have these treatments studied against all the possible species of the parasite. So these are 3 very good publications, and here there is one, and all of them have used some kind of penetration enhancer. So either ultra-deformable liposomes or DMSO or fluid liposomes. So I want to talk about our work, which is has also been centered on miltefosine. We started with this 4 or 5 years ago, and we published it just now. So here. So what have we working on? We've been putting miltefosine into liposomes, right? We tried different liposomes, and we studied cytotoxicity, parasitic inhibition in vitro, and in vivo activity, and we did also some physicochemical characterization of the effect of liposomes to try to understand the mechanism of especially of the liposomes effects. So we have worked with Amazonensis, so that adds to that list a new species that is susceptible to topical miltefosine. What we have, I will tell you only what is really struck us as strange or notable. First, that if you see here in the left-hand graph, which is parasitic inhibition as a function of concentration of miltefosine in vitro, you can see that here in this box, you can see that both miltefosine and pure fluid liposomes have parasitic inhibition properties, which was really strange. So at the lowest miltefosine concentration, we saw that the fluid liposomes per se had a parasitic inhibition capability. Also at an intermediate concentration. So this was a kind of a surprise. Although there is one other paper by an Argentinian colleague showing something similar. Then we studied all these different liposomes and these different relationships between the amount of lipids and the amount of miltefosine, and we chose, as you can see here in this arrow, we chose this formulation. So the miltefosine incorporated into fluid liposomes with a high miltefosine phospholipid molar relation. So little amount of phospholipid, high amount of miltefosine, because it had the highest efficacy in vitro. So we then went to the mice, and we got really extraordinary results. I mean, the lesion size went down until it disappeared. You can see here in the picture in the middle. This is the control on the left, and on the middle, animals treated with miltefosine or with miltefosine in phospholipids. And here is the other thing that is really strange, or not strange, but it draws our attention, because this animal here in the middle has only been treated with miltefosine 0.5% alone. So there is no penetration enhancer, no liposomes, no DMSO, no nothing. It's just miltefosine on a gel, and it cures. It's amazing. What do we need the liposomes for then, if miltefosine alone can do it? Well, when you add the liposomes, you have the same result, only faster, right? So if you see here this graph of parasitic inhibition at the end of treatment, you can see that immediately after the end of treatment, if you take the skin of the animal and you reculture it in a way that the parasites can revive, if they are still alive, if they are still viable, you can see that immediately after treatment with miltefosine, you can still find viable parasite in the skin. But if you have treated the animal with miltefosine and liposomes, you find no viable parasites. So the liposomes kill the parasites faster. Also, we studied recidiva 30 days after the finishing of the treatment, and we found that there was no recidiva in any of the animals. You can see that after 30 days, animals treated only with miltefosine do not have any viable parasites anymore. So miltefosine cures by itself. You can just use miltefosine, but if you add, and you will eventually eliminate the parasites after 30 days. But if you use the liposomes, it will be faster. So then we did some physicochemical characterization in front cells. We saw that in the top graph, you can see that miltefosine alone goes through the membrane that imitates skin faster than miltefosine that is incorporating into liposomes. So liposomes slow down the miltefosine diffusion. This is confirmed by the smaller slope in the Higuchi model. But we also saw that our liposomes are not stable in size. So they start small, 100 nanometers, but then they fuse with each other quite quickly. But this is, well, we can discuss it later, but this is, I don't think this is too important. And we found no degradation after 90 days at 4 degrees Celsius, which is also a very important thing. So our hypothesis on the liposome mechanism, why is it that the animals cure faster when miltefosine is on liposomes? Well, we think there's two reasons for this. One is that the liposomes produce a depot in the skin from where the drug is slowly released. But also we think that the lipids themselves have an effect on their own, based on our in vitro results. So I would like to mention the challenges. So this is all very nice, but of course mice skin is much more easy to penetrate than human skin. It's at least 3 times more easy to penetrate than human skin, at least 5 times thinner. So we definitely need to prove this either in animals with thicker skin or in humans. The other issue, of course, is the stability, the tropical stability of this formulation, because the liposomes have unsaturated chains, so they would easily oxidize at high temperatures. Then, of course, the great advantage of this would be that if you can keep the drug in the skin, you would have no teratogenicity, for example. But this has to be evaluated, right? So the other thing is that this would only be useful for localized cutaneous leishmaniasis. So for metastatic species of leishmaniasis, this would have to be used in a different way. So I think also of course for co-infection with HIV or some other form of immune compromise, a topical treatment would have to be used together with a systemic treatment. And in this case, I think the topical treatment would be good because it would allow, I think that we have to prove this, but I'm optimistic that it would allow a lower dose of a systemic treatment, and then maybe we could reach all of the patients. So that's all. Thank you for your attention, and these are my collaborators and the findings.